Okay, so this week we've got about 5 videos planned, like I mentioned in the intro video. So we're going to spend our time in chapter 22, which talks about speciation, and chapter 23, which is paleobiology and macroevolution. So the background is we've learned a little bit about Darwin's ideas and about how science came to recognize in the late 19th century that species are changing, and scientists started to look for mechanisms that explain how species change over time. And in microevolution, we learned about Hardy Weinberg and learned about some of the mechanisms that drive evolutionary change. So in this chapter, we're going to dig a little bit deeper into speciation, and we're going to learn about some of the processes that guide the formation of new species. Okay, so speciation is the process of species formation. And so again, Darwin kind of laid the groundwork for this, but it really wasn't until the modern synthesis, until in the early 20th century, that scientists started to really zoom in and focus in detail on how the mechanisms of evolution help create new species. And so today, evolutionary biologists view speciation as a process. Basically, it's a series of events that occur over time, usually a long period of time. And when we're studying species, we're studying the product of speciation. So we're mostly looking at species that are alive today. We can infer the speciation process in extinct species as we look at fossils, but speciation is best observed. Some of the principles we're talking about today are best observed in real time with living species. So in this chapter, we're going to look at 4 major topics. We're going to look at how biologists define and recognize species. That's number 1. Number 2, we're going to study how species maintain their genetic identity. Number 3, we're going to study how the geographical distributions of organisms influence speciation. And number 4, we're going to look at how different genetic mechanisms produce new species. So the first 2 will be the subject of this video, and the last 2 will be the subject of the next video. So let's begin by asking the question, what is a species? And so sometimes it's really easy to tell. So a dog and a cat, they're different species. Sunflowers and roses, they're different species. So we tend to think of animals and plants as being packaged in discrete units of species. So in our minds, we often draw hard lines between where one species ends and another begins. But in reality, diversity is packaged more as a continuum rather than discrete packets of species. In other words, one species might blend into another, and so we see species more along a continuum. And so in the picture, you can see these different species of ground finches. So again, kind of a classic example of speciation. So you can see all these different species of finches, and again, you can see kind of how they exist on a continuum. So they blend. There's not like just a thick-billed finch and a thin-billed finch. There's lots of intermediates in between. All right, and so again, speciation is a process, and species exist on a continuum. So how do we define what a species is? So the term species is really just a human construct, right? In other words, we use the term species so that we can have a unit of an organism that we can study. But since species are always changing, they're going extinct, their allele frequencies are shifting because they're evolving through the various mechanisms we talked about in the last couple of chapters. Just defining the term species can be difficult because species are a moving target. So here are the 3 main concepts that biologists use to define a species. So they are the morphological species concept, the biological species concept, and the phylogenetic species concept. So we'll talk about each in turn. All right, the first is the morphological species concept. So this is simply the idea that a species shares measurable traits that distinguish them from individuals of another species. So in other words, we can look at the morphology and say this is, this species has characteristics A, B, C, and D, and then this different species has characteristics D, E, F, and G, and so they are distinct species. So we're looking at mostly the outward appearance of a species or of an organism in order to determine where one species ends and another begins. So the morphological species concept is kind of the classic concept of species. This is what Linnaeus was going off of when he established his taxonomic systems. And it brings some really nice benefits, but also it has some negatives. So let's talk about the pros and cons. So for one, it's got lots of practical applications. So the morphological species concept is really helpful when we're looking at extinct organisms, for example. So you dig up fossils of various dinosaurs, and based on the bone structure, you can say, okay, we're going to define species A as having these characteristics and species B has these physical characteristics. So with extinct species, you can't breed them. You don't really know. You can't look at their genomes. You can't look at their molecular differences. And so really all you have when you're dealing with extinct species that we know from fossils, all you have is their morphology. Also in nature, just when you're out in the woods, if you're using a field guide to identify species, how are you going to tell two species of birds apart? Well, you're going to look for physical characteristics. So in the pictures on the slide, you can see at the bottom the yellow-throated warbler and the myrtle warbler. So these are both members of the same genus, but they're different species, and you can distinguish between them by looking at the different color patterns, and that's good enough for separating these two species. However, you run into some problems. So for example, if you remember in the last last chapter, we saw, remember that picture of the European garden snails, the ones that had all the different colors? They had different stripes and different patterns. Well, in that case, they're all members of one species, but they display very different or diverse morphological traits. So in that type of situation, the morphological species concept breaks down. So it also breaks down when we have two species that look almost the same. So sometimes, and you can see this in the image up here, sometimes you have individuals of two species that are morphologically inseparable. So these two species of titmouses, you can't really tell the difference by just looking at them, right? But they operate as distinct species. They don't interbreed and so on and so forth. And so sometimes the morphological species just doesn't tell us enough about the differences between species that maybe lie below the surface. And finally, the morphological species definition doesn't really tell us about the evolutionary processes that produce the species. So in other words, it's helpful to say, okay, this is one species and this is another, but because it's kind of ignoring evolution and ignoring genetics, it doesn't give us a sense of how these species are related evolutionarily. The second concept of species is the biological species concept. And so the biological species concept emphasizes the dynamic nature of species. That's from your book. Man, that's a terrible definition. Let me keep going. So a biological species under this concept is a group of interbreeding natural populations that are reproductively isolated from other groups. So in other words, a species is a population that naturally breeds with other members of the same species. And then when they come into contact with other groups or other species, they might not breed, or if they do breed, then they produce offspring that basically aren't fertile. Like, so they don't successfully reproduce new generations. So as kind of a dumb example, this is something I stole off the web. This is a giraffe, which is a species, and this is a lion. And so if you were to try to breed a giraffe and a lion, which would be an interesting experiment, you'll find that it's not going to happen. So the lion and the giraffe aren't going to breed. They're not going to produce offspring. And so here we can safely say that they are distinct species. Now, of course, the morphological species concept would help here too. You'd say, look, the giraffe and the lion don't have any morphology, a lot of morphology in common. So it's also going to distinguish these as separate species. But here we're defining them as two species because they're not going to interbreed. Even though they live in the same habitat, they're not going to interbreed and they're not going to produce offspring. In the lower picture, you see that these are two giraffes that look morphologically, to me, they look the same. And so are they members of the same species or different species? Well, when you put them together, if they breed and if they produce a successful offspring that can also breed and produce offspring, then you would say these are members of the same species. So the biological species concept notes the genetic cohesiveness of a species. In other words, populations of the same species experience gene flow, and this gene flow mixes their genes and their alleles around and creates diversity, but at the same time allows them to continue to interbreed together. So a species is basically one large gene pool. So there might be local populations, but because they're all interbreeding and the genes are flowing between individuals of this population, we would call them one species. The second part of the biological species concept emphasizes the genetic distinctness of each species. So these species will be reproductively isolated. That means they can't exchange genetic information with other species or other populations. So we're also going to focus under this concept on how the gene pool of a population or a species basically mixes genes within that gene pool or population, but it doesn't mix genes with other populations or other species. So they remain cohesive within the species and they remain genetically distinct from other species. So this is helpful. We can talk about pros and cons. This is helpful in the sense that this explains why members of one species look alike. Why do they look alike? Why will their morphology often be very similar, like we see with the giraffes below? And that's because they're all sharing genes. And so they don't have as many, because they're all members of one species, they share a lot of the same genes. And because they're sharing the same genes, then their morphology is going to be pretty similar. A con of the biological species concept is that what do we do with species that maybe produce asexually, right? And so if we're depending on their ability to breed and produce viable offspring, right? If a species is reproducing asexually, then they're not going to breed, right? They're just going to make clones of themselves and give birth to them. So it's not helpful in asexual populations. So that would include some protists and bacteria and fungi and plants and some animals as well. So similarly, when we're studying extinct organisms, like we talked about just a minute ago, if we're looking at fossils, we don't know if these different fossils these organisms, if they were alive, if they would breed together or not. We don't know that because they're dead. We're just looking at their fossils. So it's not really, it might be helpful, this concept might be helpful with living species, but it's not necessarily helpful with extinct ones. And also, sometimes it's just not practical. I mean, so going back to this giraffe and lion definition or example, it's kind of silly to try to breed a lion and a giraffe to see if they reproduce, to see if they're members of the same species. In other words, a lot of times morphology provides really good clues, and so we can easily say, look, these are different species, and we don't have to, practically we wouldn't try to get them to mate because that's kind of weird. The phylogenetic species concept is a concept that uses morphological and genetic sequences to reconstruct the evolutionary history of an organism or of a species. So basically, in this concept, we're looking at species and defining them as ones that share a recent evolutionary history. So the phylogenetic species concept builds evolutionary trees and says, okay, this is a species and this is a different distinct species, and that's because they've separated in evolutionary time. So an advantage of this is you can look at anybody, you can look at any living species, or even extinct species, And if you can create their evolutionary history, you can create their phylogenetic trees, then you can distinguish one species from another. Um, you can also apply this concept to asexually reproducing organisms. So in other words, you don't have to worry about breeding. You're just looking at how their evolutionary histories diverge. Um, Now, a problem with it is that for some species, we don't have a good evolutionary history. So if you're lacking good evolutionary data, then you're going to have difficulties figuring out where one species ends and another begins. So this is very much dependent on evolutionary analyses. So it's good in the sense that here we're not just concerned about, making a field guide and saying, well, this is one species of bird and this is another. It's adding a layer of detail where we're figuring out, okay, this is a species and this is a species, and we know this because here's their evolutionary history. But well, oh man, I just lost my train of thought. So this concept is good for practicing or looking at species through the lens of evolution. But again, if we don't have good data on evolution, then it's going to cause problems. We're not going to have robust enough data in order to um, track their evolutionary history and therefore define different species. All right, so we've again, I've talked about species as if this is like a concrete idea, but there's lots of gray areas. So let's look at a few examples where we can split up populations, not just as a species, but as subspecies and ring species and clines, as you'll see in the next slide. So the first concept to talk about is subspecies. Subspecies are just local variants of a species. So if you look at the example on the left, we've got some subspecies of rat snakes. And overall, you can see that they live in eastern North America, right? So here's their range. But if you zoom in and you look at them up close, you see that the rat snakes living up here tend to be black, and the ones that are down here tend to be yellow, and the ones down here are gray, and the ones down here are orange. And so if morphologically you might say, you know what, these are different species because they have different colors. Evolutionarily, you might say, well, they all share a recent common ancestry, or phylogenetic species concept. You might say, hey, look, they share a recent evolutionary history, so maybe they're all one species, or well, maybe they're two, just depending on your analysis. And in the biological species concept, you might say, well, can they interbreed? And the answer is yeah. So if you take a black rat snake and you put it in a cage, and you shouldn't put a snake in a cage, it could definitely escape. So you put them in a terrarium, like a glass aquarium, right? Well, it's not an aquarium because there's no water. You know what I'm saying? And the black rat snake might successfully breed with the Everglades rat snake and produce viable offspring. So in the biological species concept, you might say, they're members of the same species, right? But at the same time, one's black and one's orange, or one's black and one's yellow. So there's morphological differences that need to be explained. So subspecies are basically just one level or taxonomic layer below species. So we would say, yeah, these are all members of the same species because they can interbreed, for example. But because they exist as different variants or different forms, let's go ahead and put them into different subspecies. So if you look at the binomial nomenclature, in other words, the genus and species, this E is representing the genus. This, what is that? An O is representing the species. And then this one has a subspecies label, obsoleta. This one, same genus, same species, but it has a different subspecies name, which is Rossellini. All right, that's subspecies. Ring species are similar. All right, so you can have subspecies that kind of form a ring. And you can see that in the example of these salamanders. So ring species are adjacent populations that can exchange genetic material, but the gene flow occurs only through intermediate populations. So let's go to the example. So again, we've got all these different salamanders throughout Western California. And let's start with the painted salamander. So it lives up here off the coast, or off the coast of California near the border of Oregon. And it has some distinct morphologies. And if you go way down here to this salamander, the large blotch salamander, you can see it lives in Southern California, and it looks different. And so if you were to drive this one up here and put it in a terrarium, they might successfully breed and have offspring. But that's you driving the salamander from one, spot in California to another. So what's the likelihood that that's going to happen naturally? And in reality, it's pretty low. In other words, the large blotch salamander is going to rarely, if ever, come into contact with a painted salamander. So we might say that all of these salamander species form a ring. So you can see that that this population is fairly distinct, and it might encounter the Oregon salamander and they might interbreed, and the Oregon might encounter the yellow-eyed and interbreed, and the yellow-eyed might encounter the Monterey and interbreed, and the Monterey might encounter the large blotched and interbreed, but the large blotched is going to rarely interbreed with the painted. So are these different subspecies or different species? Right? In other words, because they can interbreed, does that make them members of the same species? Or because they don't in nature naturally interbreed, does that make them different species? So by labeling them both subspecies and ring species, we can signal that this one species of salamander exists as different forms that rarely interbreed. Right? They might interbreed with their neighboring subpopulations or subspecies, but overall the gene flow isn't really flowing directly. The genes aren't flowing from here to here. The genes would have to flow here, and then here, and then here, and then here. So ring species are just a way of keeping track of that. So again, you can see lots of gray area exists where one species ends and another begins. And also I want to note that since speciation happens over long periods of time, you could say that with subspecies and ring species, that this is speciation in progress. So in other words, if we were to fast forward the arrow of time and come back, I don't know, 10,000 years from now, it might be that the painted salamander and the large blotch salamander, because of their genetic differences, if you put them into a terrarium, they won't interbreed. So in other words, by looking at subspecies and ring species, we can see speciation in action. Clines are similar. So a cline is a measurable gradual change over a geographic region in the average of a phenotypic trait. And let's look at this example. I think this one's easier than the one in the book. So this is deer, white-tailed deer, and you can see that they live in northern Canada and the lower 48, and they live down in Central America as well. And if you were to look at one of these white-tailed deer from Central America, it's going to be small and have small antlers. And if you were to look at a deer in Michigan, you'll see that it's much larger and has larger antlers. And so as we move from north to south, we're going to see a gradual change in the average antler size and body size and skull size of the deer. So we would say that these are clines, right? And so they're exhibiting clinal variation. So why is that? It's because the deer that are living in South or Central America, it's hot and it's sunny and you've got long seasons and there's totally different plants for them to eat and predators and so on than compared to the ecosystems and the environment in Michigan. And so the differences in geography lead to differences in the environment, which lead to different phenotypes having different levels of fitness, right? And so that gets expressed as different phenotypes depending on where you are in the geography. Does this mean that these are a different species than these? Well, no, because again, if you bring them, if we're going by the biological species concept, if you bring them together, they can successfully mate and reproduce. Under the morphological species concept, you might say that they're different species, or you might just say, well, no, they have different morphologies, but it's because of the geographic variation, so they're clines. So in other words, a cline, much like subspecies and ring species, is just a way to communicate that there are different phenotypes over the natural range of an organism. Okay, next section is maintaining reproductive isolation. And so if we're going off the biological species concept, then we're concerned with the ability of two populations or two species to mate and have viable offspring. So what are some ways that separate different populations in ways that prevent them from reproducing successfully and having viable offspring? And one answer to that is reproductive isolating mechanisms, which are just mechanisms that prevent individuals of different species from mating and reproducing successfully. All right, so in this example, which I took off Google, we start with one species of fruit fly, and over time, in a laboratory setting, they split this population into two, one and two. And in this one, they give it one type of food, and in this population, they give it another type of food. And so this population interbreeds with these. They're all eating this food and the same here. And over time, then the ones that are eating this food kind of become their own population, and the ones eating this food become their own population. And then if you were to reintroduce these two populations, you might find that the ones that ate the yellow food just interbreed, and the ones that ate the purple food just interbreed. And so we would say that these two populations are now reproductively isolated. In other words, they might not mate at all, or they might mate but be genetically distinct enough that they don't produce viable offspring. Either way, during this time up until the end, right, in all three of these points, these two populations are reproductively isolated. Here, they're isolated because they're separated. They're living in different, vials of the experiment in the experimenter's lab. And then here, when you put them in the same vial, they're still reproductively isolated. They're not breeding with each other, or at least they're not breeding successfully. So this is an example of reproductive isolation. Sometimes we find that when two species that exist, if they come together, they might actually mate, right? But as a result, they'll end up producing a hybrid. So a hybrid is just an offspring that has two parents, and each parent was a member of a different species. A real life example is a mule. So a mule is a hybrid of a horse and a donkey. So are horses and donkeys members of the same species or different species? Well, they can successfully breed. So in the biological species concept, we might say, well, doesn't that mean that they're members of the same species? The answer is still no, because what they produce, a mule, is infertile. So you can breed a horse and a donkey and you can get a mule, but the mule has a set of chromosomes that basically allows it to, or prevents it from, having viable offspring. So if you mate two mules, then the female isn't going to get pregnant. She's not going to give birth to another mule. So a hybrid is kind of this weird, they're kind of stuck. They're this weird combination of two different species as their parents, but and they might be their own species, but they're not a functioning species. They can't reproduce on their own. So hybrids are just a weird, semi-successful product of two different species that mate. All right, so we can split reproductive isolating mechanisms into two subcategories: pre-zygotic isolating mechanisms and post-zygotic isolating mechanisms. So you probably remember what a zygote is. So if you look at this image, is this from your book? Nope, stole it off Google. So if you look at this image, we've got a gamete from individual 1 and a gamete from individual 2. So maybe this is a sperm and this is an egg. So when the two gametes fertilize, when they fuse together, they produce a zygote. And normally that zygote is going to start undergoing mitosis, and its genes are going to are going to oversee its development, and that zygote will eventually develop into a viable offspring. So pre-zygotic isolating mechanisms prevent the zygote from forming. So they prevent the sperm from reaching the egg, all right? Or they prevent the genetic information from one species from interacting with the genetic information from another species. We'll talk about some of these barriers that you see here in just a minute. In post-zygotic isolating mechanisms, the two species or the two individuals might breed and they might produce a zygote, but in post-zygotic isolating mechanisms, that zygote, for whatever reason, doesn't develop into a viable fertile offspring. So there's three types of post-zygotic barriers that we'll talk about shortly. So first, let's talk about these. Let's talk about prezygotic. Actually, first, here's a table. I'm sorry, the top of it's cut off, but here's a table that is that lists all these different mechanisms together. So there's 5 mechanisms of prezygotic mechanisms, prezygotic isolating mechanisms: ecological isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. So this table kind of summarizes those nicely. We'll talk about each of them in turn. Post-zygotic mechanisms, there's 3 subcategories: hybrid inviability, hybrid sterility, and hybrid breakdown. So this is just a nice table for you to come back to when you're studying. Again, we're going to talk about each of these in turn. Let's talk prezygotic. So prezygotic isolating mechanism number 1 is ecological isolation. So here you get 2 populations that are separated ecologically. And so over time, because they're separated ecologically, they become distinct or separate species. So let's start with lions and tigers. So lions live in the open grasslands of the savanna, currently in Africa primarily. And then tigers prefer to live in forested ecosystems, like in Siberia or India. And so in captivity, lions and tigers can interbreed and produce a hybrid, which I'll talk about that more in a minute. But in nature, because they're living in different ecosystems, and these ecosystems aren't next to each other, in other words, the savannas of Africa and the forests of India are pretty far away, and there's deserts in between. So it's very unlikely for a lion to migrate to India, just like it's unlikely for a tiger to migrate to Africa. So because lions and tigers are separated ecologically, then they are prezygotically isolated. So it's very unlikely that they're going to breed and that they're going to produce a zygote. Another example is chipmunks. So if you look at this picture, you can see that there's 4 different species of chipmunks. This is in the Sierra Nevada mountains of California. So the alpine chipmunks live up here, lodgepoles live here in the lodgepole pine forest, the yellow pines live here in the yellow pine or pinyon pine forest, and then the least chipmunks live down here in the sagebrush deserts. So each of them inhabit a different ecosystem, and because of that, it's very unlikely that the alpine, even though these ecosystems are next to each other, it's very unlikely that the alpine chipmunk is going to move downhill and move into this ecosystem and mate with a lodgepole, just like it's unlikely that lodgepole chipmunk's going to move uphill and move into the alpine zone and breed with the alpine chipmunk. So again, because they're inhabiting different ecosystems, they're ecologically isolated. So over time, let's say one population of, or one species of chipmunk initially moved in. Over time, the subpopulation of chipmunks that are up here didn't breed with the ones that were here, and these didn't breed with here, and these didn't breed with here, and so on. And so therefore they were ecologically isolated, and you don't have gene flow between the populations that are inhabiting these different elevations on the mountain range. And that leads to speciation, right? So again, in both cases, we get a new species of a lion and a tiger forming because they're ecologically isolated. And here we get 4 species of chipmunks that are forming because they are ecologically isolated. Temporal isolation. Temporal isolation has to do with time. Here you get new species forming because they're separated temporally. They're separated in time. The best example of this is spring field crickets and fall field crickets. So spring field crickets are adults in the spring, and they mate and reproduce and produce offspring in the spring, and then they maybe overwinter as eggs or nymphs. And then, well, probably as nymphs. And then the fall field cricket, it hatches in the spring, and the adults develop in the fall, and so they mate in the fall and overwinter. Man, I should know how they overwinter. I'm guessing they overwinter probably as eggs. But anyway, the point is, these two populations of crickets are temporally isolated. So the adults of the spring-field cricket aren't going to encounter the adults of the fall-field cricket, so they're separated in time, or they're separated in different seasons, and so they're not going to produce a zygote because the adults aren't going to interact with each other. So again, this is just how two different species of crickets can form over time. It's because they're undergoing temporal isolation. In behavioral isolation, you get species forming because they're separated through behavior. So two examples. Let's start with the one on the right. This is confusing at first, but this is different patterns of fireflies. So in fireflies, the males and females attract each other or signal to each other by blinking lights that are produced through a biochemical reaction in their abdomen. And so this species of firefly, the individuals, I'm guessing that usually it's the males, will fly and they'll blink, 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 fly, and then drop down, blink, 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 fly up, drop down, blink, blink, blink. Let's go to these guys. This species of firefly will blink, will fly, and will blink, 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 blink. And so these fireflies become separated because of behavior. So these males over time are going to attract, or are going to get responses from females that are attracted to this light pattern, whereas these fireflies, the males are going to attract or be communicated with by females that are responding to this light pattern. And so the differences in behavior lead to a separation of the individuals in this population, right? They might be in the same ecosystem, they might be alive at the same time, but the differences in behavior keep genes from flowing between these two subpopulations, and over time they become distinct species. The second example is the birds of paradise. So the birds of paradise are well known for different courtship rituals, so different feather patterns and different dances and different songs. And so over time, the females or the males of one group are going to do a particular dance with particular color patterns, whereas the males in another group are going to do a different dance with different color patterns. So much like the fireflies, some females will be attracted to this dance and this color pattern, and other females will be attracted to this dance and this color pattern. And so over time, the differences in courting behavior lead to different subpopulations, and over a long period of time, that gives rise to different species of birds of paradise. The fourth type is mechanical isolation. So in mechanical isolation, basically you don't get zygotes forming because of different mechanics, differences in reproduction. So let's actually start with the dog example. So this is a Great Dane and this is a Chihuahua. So they're members of the same species. Let's say that you have, a population of Great Danes running around and a population of Chihuahuas running around. Well, they're in the same ecosystem, right? And temporally they're not isolated, so they're able to mate during the same time period. And maybe the behaviors are the same. In other words, there's no particular, lights flashing or courtship rituals that they're doing. So you'd think, well, maybe they'll interbreed and Chihuahuas and Great Danes will become more similar, and these subpopulations over time will lead to one population of dog. Right, but that's not going to happen because of mechanical isolation. So in other words, the Great Dane is really big and the Chihuahua is really small, and so they're not going to interbreed just because it's going to be hard to mate. Right? And so they're going to remain mechanically isolated. So the Chihuahuas are going to breed with other Chihuahuas, the Great Danes are going to breed with other Great Danes, and so these subpopulations over time might become new species. An example from the book is in monkey flowers. You've got purple monkey flowers and scarlet monkey flowers. And if you recall, plants produce flowers to attract pollinators and move their pollen from one to another. That's going to produce the zygotes. In the purple monkey flower, the way that their pistils and stamens are arranged are going to attract particular pollinators. Sorry, I'm looking up in the book. Where is this? Sorry. So, so this one attracts hummingbirds, and this one, the scarlet monkey flower, attracts, where is it in the book? Bees. All right, so in other words, the differences in, that can't be right. Right? This is so embarrassing. I need to be able to edit these so I don't make a fool of myself. Purple monkey flower, pollinated by bees. So I did it backwards. Golly, sorry. These are pollinated by bees. These are pollinated by hummingbirds. My bad, guys. And so that's because the differences in nectar availability, differences in color, differences in flower structure. And so because they have different pollinators, then they're mechanically isolated. A bee landing on this one isn't going to pick up pollen. And a hummingbird that's drinking nectar from this isn't going to pick up pollen. So over time, you get two different species of flowers just because the pollen isn't being transported between these two different flower structures by their pollinators. I don't know. That was a disaster. I'm sorry, guys. And the last category is gametic isolation. So sometimes you might have individuals of different species that mate. Right? So in other words, that maybe they're not separated by ecology, or they're not separated by time, or they're not separated by behavior. So maybe they'll be attracted to each other and they'll actually mate. However, in gametic isolation, when the sperm fertilizes the egg, right, the gametes aren't compatible. So the sperm might swim to the egg, but it's unable to actually fertilize it and produce a zygote. So that's fairly straightforward. They're able to breed for whatever reason, but they don't actually produce a viable zygote because the gametes are incompatible. Maybe the sperm doesn't fertilize the egg for whatever reason, or maybe they have different chromosome numbers or whatever. All right, post-zygotic isolating mechanisms. So in these examples, you have individuals that can maybe actually breed. Again, maybe they're taking up the same ecology, living in the same environment, or maybe there aren't behaviors that separate them and prevent them from breeding. However, and the gametes are even compatible, so the sperm will actually fertilize the egg and create a zygote. However, in these cases, the zygote, for whatever reason, doesn't end up developing and producing a viable offspring. So let's talk about each of these in turn, and I might return to some of these pictures. Oh, snap. I don't have, daggone it. Sorry. I thought that I had slides for these. I don't. All right, let's talk about each of these in turn on this slide. So first up is hybrid inviability. So in hybrid inviability, the two individuals breed. The example from the book is a sheep and a goat. So they're able to mate. The sperm is able to fertilize the egg. A zygote is formed, but then there's post-zygotic isolation. In other words, that zygote forms, but because the hybrid that's produced, the hybrid that the offspring that would be a hybrid of a sheep and a goat, it doesn't actually develop. So whichever one got pregnant, the sheep or the goat, the zygote forms, but then it dies. So there's enough genetic separation between sheep and goats that the zygote doesn't develop and it does not produce a hybrid. So we'd say this is hybrid inviability. In the second example, hybrid sterility, you might get two species that mate and the gametes fertilize and produce a zygote, and the zygote develops and the female actually gives birth to a hybrid, but the hybrid is sterile. And so this goes back to our mule example. The horse and the donkey mate, a zygote is formed, and they give birth to a mule. However, that mule is sterile. The mules can't mate with other mules and produce offspring. So here it's kind of a dead end. right? The zygote develops into a mule, but the mules can't reproduce, and so you're not going to get a new species forming from the hybrid, and therefore genetically the horses and the donkeys remain distinct populations and therefore distinct species. The third example is hybrid breakdown. So in hybrid breakdown, you might have gametes fertilizing, you get a zygote. The zygote can develop, so there's not hybrid inviability, and then the hybrid is able to reproduce, so there's not hybrid sterility. However, over time, the hybrids exhibit reduced survival or fertility. So in other words, the hybrids are less fit than the parents. And so this could be because there's chromosomal abnormalities or there's harmful genetics going on. The point is the hybrid breaks down and it's not as fit as the two distinct species that mated in the first place. I hope all that made sense. If not, see the discussion board or talk to me in Zoom. Just so I cover all the pictures in the slide, here's a lion that's crossed with a leopard, a female lion, a male leopard. They are able to reproduce. All right, this is going to happen in captivity. And actually, a lion and a leopard aren't separated ecologically. They both live in the savannas of Africa. They're not separated temporally because they are around at the same time. They're not necessarily separated behaviorally. So in other words, they don't have distinct courtship routines that are going to keep them from mating. And they don't have gametic isolation. So in other words, the sperm from the leopard will fertilize the egg from the lion and produce a zygote. They don't have hybrid inviability. In other words, the zygote will go on to develop and produce the offspring, which is called a leopon. However, the leopon is sterile. And so you mate it with another leopon or something else, and it's not going to be viable. In this picture, we have zebroids. So a horse can breed with a zebra, but the zebroids are usually sterile. So another example of hybrid sterility. All right, so hopefully it's coming together. So hopefully you see that these reproductive isolating mechanisms help prevent gene flow between two populations, which over time keep those populations isolated. In other words, it keeps them distinct. And these various mechanisms help ensure that those two populations in time become distinct species, and that those distinct species will have different morphologies, they'll have different phylogenies, and they will have different levels of reproductive success, or they'll have different levels of reproductive incompatibilities, maintaining them as distinct species. All right, see me in the discussion board for clarification. Wasn't the best lecture in the world, but we're online. Don't have many options. Hey, staying positive. This is awesome. See you next time.